move into it. So where we kind of ended was looking at the anaerobic processes of pyruvate. And why do we do our anaerobic processes? What was the whole point of those? To make our NAD+. Right? So we need that to effectively restart glycolysis so that we can produce some energy. Because we can't get enough energy, or we can't get energy through citric acid cycle, because that's ultimately leading into oxidative phosphorylation, which requires oxygen. Okay? So if we're anaerobic, we don't have the oxygen. So one of those dead ends is what happens in yeast that we take advantage of in the form of alcohol and, for that matter, carbonation. And so that process was taking pyruvate and turning it into acetaldehyde and then later into ethanol. Okay? And that goes through our pyruvate decarboxylase. And if we look at what changes to our structure, when we look at pyruvate and we compare that to acetaldehyde, what happened? It lost a carbon and an oxygen and a second oxygen, so we lost CO2, and at the same time gained a hydrogen. Okay. Is this a redox reaction? Yes. Okay. It's a little bit tricky. If we look at our carbon where the chemistry is happening, we are losing a carbon and replacing it with a hydrogen, which is more electronegative, carbon or hydrogen. Carbon. So we're losing an electronegative element at the red carbon and gaining a less electronegative element. That is the definition of reduction. Okay? So we are doing a reduction reaction, which means we would need a redox mediator, and our redox mediator, our NAD plus or NADH. Which one would we need? NADH, because the NADH is what's supplying that hydrogen. Officially our hydride in this case, because our other group kind of runs off with our electrons, theoretically. Okay. This is a pretty neat reaction, and we'll see a mimic of this in the citric acid cycle, okay, or getting into the citric acid cycle, called pyruvate dehydrogenase, doing effectively the same thing, just not going all the way to the acetaldehyde. Okay? Taking it to the active form of the acetaldehyde, which would be this piece known as I ran out of space. I can go the other way. Why? Yeah, and Jen already said it. Let's see if I can spell it correctly. Did I spell that right? It's not an acetal, it's the acetyl. Okay? The acetyl group is saying that that carbon, whoops, this carbon here can then be linked to something, okay? And that piece is known as an acetyl. Okay? That's where we're getting acetic acid from, because you add an OH at that position and it's now a carboxylic acid, so it becomes an acetyl carboxylic acid or acetic acid. Okay? So our dehydrogenase keeps that active. Why is that not active right now? It's an aldehyde. Okay? We've kind of finalized that structure. If we want that acetyl group to be able to be reactive, I'd have to remove that hydride and then have it react. Okay? That's an extra step. Okay? So if we're looking at the dehydrogenase reaction, we won't go all the way to the acetaldehyde. We'll go to the acetyl group and then have something attached that we could easily remove. A carbon-hydrogen bond is not an easy bond to break because I talked about it a lot with my OChem students, less so with you guys. Why is a carbon-hydrogen bond one of the harder bonds to break?
There's not a big partial...
Not a big partial difference in charge, meaning we have a nonpolar covalent bond, and our nonpolar covalent bonds are the strongest bond. Okay? So that becomes a very difficult bond to cleave. So to initiate this whole process, we end up using a cofactor called thiamine pyrophosphate to be specific, though the thiamine is actually the one that's doing the chemistry. The pyrophosphate, okay, or really your phosphate groups, makes it TPP, are there to make sure the molecule can grab a hold of it. Okay, those are effectively your handles. And then alcohol dehydrogenase does the next step. But I don't care about that right now. I care about our thiamine. So if we take a look at our thiamine, we've got vitamin B1 shown up in kind of the middle there. And if we make that thiamine pyrophosphate, all we've ended up doing is adding some phosphate groups. Those phosphate groups make it more charged, which means it's going to be easier for an enzyme to go ahead and grab a hold of those. There's a big reason why in the thiamine they showed that red section highlighted out. What do you think? Where do you think all the chemistry is happening? It's happening at that red section. Okay. What chemistry do you see?
Charge.
Okay, charge where? Nitrogen is positively charged. Okay. What does that mean?
It wants 3 electrons.
It does not want to be positively charged, so what could it do? Steal electrons from...
CH2.
So we could try and do an elimination and take electrons that way. There's an easier source of electrons. How about the pi bond? What does that then make that carbon? Makes it a carbocation. We have a positive charge kind of chilling out there. So when we look at thiamine, that carbon is relatively electrophilic, okay, which is kind of neat. Okay. What else could potentially happen? And this is where things get all sorts of weird.
Aren't there lone pairs of the sulfur?
There are lone pairs on the sulfur, and sulfur in general just does nasty, weird stuff. It is not a pleasant not a pleasant atom to work with, because pretty much all the rules that we use in OChem for explaining where electrons move and how they move are completely irrelevant to sulfur. And sulfur does whatever the frick it wants, which makes it a real pain in the butt, which is actually probably one of the reasons why this structure does this chemistry. So that purple hydrogen has some interesting characteristics. What do you guys think? It's actually acidic. Okay. If it's acidic, what happens? It gets removed, and in the process, what do we have? A lone pair of electrons on that carbon. Which means the carbon would act as a nucleophile. Wait, what did I just say it acted as? An electrophile. Okay. So thiamine, if it acts as an acid, unloads that hydrogen and it becomes a nucleophile. But due to that nitrogen being positively charged, that carbon can also act as an electrophile. And this is where things go really weird. We have a single atom acting as both a nucleophile and an electrophile. Has that ever happened before in, say, your OChem background? Probably, good answer. Okay, it actually happens in a couple cases. Okay, if we look at a standard alkene, and we looked at, say, Br2, what happens in this reaction? Pi bond comes out, attacks the bromine, which then kicks out that bromine, and then
Substitution reaction?
It's not a substitution, because if we did that, what happens? We've lost the pi bond. Okay? And we could end up with something looking like this. Okay? And what should be a giant red flag is we don't get that. Okay? And this is where it would be an OChem review. What structure do we get instead of that carbocation? Anybody remember?
Doesn't bromine add across the, so bromine that the carbon add.
Carbo, by definition, means it's a carbon that's positively charged. We end up with this structure. Okay, or at least we have evidence to support that this is the case. Another lone pair there. Okay. Which is a really weird structure because
The bond angles.
bond angles, bromine doesn't like to have two bonds as well. I mean, everything about that is weird, which is why that molecule is now super reactive and the second bromine can come in and react with it. Okay? But if that's what I made, are my arrows correct? The red curved arrows? No. Because I formed one of those bonds, yes. That's one red arrow. The second red arrow says I have bromide, Br minus, floating around. I have not explained the second bond. How do I get that second bond? And we've got a purple bromine. That purple bromine is the one that forms the bromonium ion. It gained electrons and lost electrons. What charge would it have? Based off of our arrows shown so far. Gained and lost. It would stay the same. Okay. What charge did it start? Neutral. According to our product, that charge must be, so what has to happen? It'd have to lose more electrons. Where is it losing those more electrons? The other carbon of our pi bond. And we form a bromonium ion. What did bromine act as in this reaction? An electrophile and a nucleophile. It's doing exactly what the thiamine's doing. Okay. Why could bromine do this? Okay. And then this is probably pushing the limits of what you learned in 230. Those of you who took the full year should be able to answer why bromine does this. Bromine is massive. So we form an unstable carbocation intermediate, but due to bromine's size, it can kind of lean over, stabilize that carbocation, and form that three-membered ring. It's only because bromine is large. What is the issue about our thiamine carbon that we said can be both a nucleophile and an electrophile?
It's an organ structure.
What's the atom? What's the size of carbon versus bromine?
Small.
Small. Would we expect carbon to be able to do that chemistry? No. Okay. Because of that size. Well, it turns out that we have still seen carbon having done this chemistry before. We have seen the formation of this structure. That is a cyclopropane. 3 carbons in it. It's formed from an alkene. How do we get that to happen? Oops. We need lone pair electrons, but then we also need something to cause that carbon to be positive. Why does attaching bromines to it make that carbon positive? Bromines are very electronegative. That species, if we look at, and this is everybody's favorite question, that purple carbon, can you tell me its hybridization?
That's what?
It's hybridization.
That's a word.
Yes. That should have been explained in any OChem class. That doesn't mean you remembered it. It's okay. The hybridization of that carbon, it has 3 groups of electrons, which means it is sp2. What is its formal charge? So, valence electrons, 4, minus number of bonds, 2, minus non-bonding electrons, 2. It's neutral. So it has a lone pair of electrons, and a lone pair of electrons could act as a nucleophile. It also has an empty p orbital. What could you put in an empty orbital? electrons. The carbon can act as an electrophile as well. That species is not easy to make. Okay? It is so difficult to make that it gets its own special name. It's known as a carbene. Okay? Carbene chemistry, depending on how we make it, is not easy. Okay? And usually super rare to the point where very few people talk about it in OChem. Right? Though it is technically part of the curriculum in the year. I don't know if it shows up in the one semester version or not. Right? That molecule is now neat because we have an electrophile and a nucleophile that can do all of our chemistry. But we had to have special conditions to get that to form. Well, what is thiamine doing? The exact same thing. It's making a carbene. It's the biological equivalent of a carbene. That's pretty fascinating, at least to me. Right? We're taking something that it was so insanely reactive in OChem that we could barely ever form it. One of the common ways to form it is using a reagent known as diazomethane. Okay? Diazomethane is so reactive that the glassware that you use to do the reaction in cannot have powdered glass. You've seen the joints on glassware being powdered. If it is powdered, that causes enough friction on the glassware that it will combust. Yeah. Your glassware has to be perfectly smooth and polished. If it has like a, you know, you take a glass rod and you scrape to get a crystallization and you get a cut kind of through the glass, that can cause the diazomethane to combust. Just the presence of that causes it to combust, not the action of making it, the presence of it. So it is an insanely dangerous reaction. People lose fingers when working with diazomethane. Okay? So kind of neat. Okay? Such a reactive thing, and yet, what does biology do? Biology runs that same reaction, okay, using thiamine, an important cofactor for individual reactions, because we need that nucleophilic and electrophilic characteristic. Okay? So that's what this next slide, which is officially like 4 or 5 slides later, for those of you that printed it, I just shifted it around. Okay? To get our carbene, so if we take a look, that's our reagents. We'd start with bromoform. I think I got that right. Okay, so a carbon with 3 bromines. We expose it to a strong base. What does that strong base do? Removes the hydrogen, and we'd be left with carbon with a lone pair and now 3 bromines. Because bromine is an obnoxious jerk, what can it do? It can steal the electrons. This would take us to, come on, pen, our active carbene structure. That carbene can react with the alkene, and we'd form our cyclopropane structure. Okay? So we needed a lot of things to get this to work. We needed the halogens to help draw electron density away to make that hydrogen acidic so that we could actually have the base react. We needed the halogens to act as a leaving group to take electrons away from the structure so that we could actually form the carbene. So lots of kind of perfect situations happen to get that to work. Right? And yet, well, since you're drawing, I'll pause. Because when I hit the next button, it all deletes. Right? And yet, what does thiamine do? The exact same chemistry. Right? Or the overall chemistry of getting a carbon to act as both a nucleophile and electrophile. And that is pretty freaking cool. Right? Thiamine here is rotated, just so you know, in case you missed that. And this structure has another issue because it doesn't show the positive charge on that nitrogen. But that is our carbene equivalent of our carbon. That hydrogen is acidic enough that it can get removed. The electrons can go to the carbon, and we now have that carbon acting as both a nucleophile and an electrophile. Okay? Pretty freaking cool, in my opinion. Okay? We have a stable carbene in thiamine. Okay? That still needs to get processed, so we're talking about pyruvate. So pyruvate is shown in the upper left-hand corner reacting with our thiamine. Okay, so here's our pyruvate structure, okay, or at least the similar comparison of it. Okay. Officially, that R1 has that acid, has that carbonyl on it. The thiamine will attack the ketone form of our pyruvate. think this goes back to the beginning. We can then do some acid-base reactions. Notice that it acted as a nucleophile, and then at a later stage, what ends up happening, the electrons come in, and it accepts electrons, making the carbon act as an electrophile. Okay. That kicks off our R1 group. Which remember, we said the R1 had the carbonyl attached to it. So what are we actually kicking out? CO2. There's the decarboxylation step of our reaction. We then have to cleave off all those extra pieces that we put on there, our R2 and the rest of the carbonyl. That's where we do the reverse reaction of that whole process. And we can end up kicking out the aldehyde, and then we regenerate our thiamine at the end. Okay? Kind of a neat process, okay? At least to this person. Okay? At the end of class on Thursday, Chris came up and said, hey, I just wanted to say this is actually really kind of neat, because if we consider what's happening here, this reaction is happening probably billions of times in your body, okay, every second. All of those arrows, all that pushing, all that stuff is happening that fast. This is not a slow time scale. These things are happening insanely fast and insanely effective at their process. It's kind of fascinating to realize the complexity of what's happening. Okay. Which kind of ends us with this chapter, or this, yeah, I think it's this chapter. If we look at glycolysis, the overall process is exothermic, releases about 73 kilojoules per mole. Okay. We get our glucose converted down into 2 pyruvates. Okay, that was all our structure stuff. That 73 kilojoules per mole that was released is enough to convert 2 ADPs into ATP, because ATP has, what, 32? Or is it 31? It's got to be 30, no, 32, I think. Okay. So 2 times 32 gets us 64. We've got about a 10 kilojoule excess, so we will end up producing energy, but it's enough, or heat energy, but it's enough to go through and actually form our ATP. Okay. If that process was not exothermic, would we be able to produce ATP? No. Okay. ATP is a higher energy molecule. Glycolysis has to be exothermic. Okay. Or as I believe it was Anna that asked about it, exergonic is the same thing as exothermic. The gonic part, for some reason, is never stuck in my head. Probably because I'm a chemist. Okay. That's the end of glycolysis. Yay! Officially, if we were to mimic ASU's biochemistry program or biochemistry class, we would then move into gluconeogenesis, which is the reverse process. Okay, gluco for glucose, and then neo, new, genesis, making. So how do we then take the products of, say, pyruvate or other molecules and reverse that process back into glucose? Okay, if in case we needed energy and we didn't have any food. Okay. For whatever reason, I don't do that. So maybe we'll talk about it at the end of this unit. Depends how far into citric acid and electron transport chain we get. I just feel that those are kind of more important and more commonly tested on than gluconeogenesis. Okay. So, citric acid cycle. What are we starting with? Pyruvate. Okay. Citric acid is kicking off from glycolysis. Okay. So, glycolysis and a bunch of other things go in to make acetyl-CoA, okay, which we haven't officially talked about. So glycolysis ends with pyruvate. That's our first line. Okay. Pyruvate does not directly go into the citric acid cycle. Okay. What we end up doing with pyruvate is getting rid of a CO2, just like the alcohol dehydrogenase, okay, to form acetyl-CoA. Okay. Why the acetyl-CoA? Well, what does coenzyme A do? Transfers acetyl groups. So what we're trying to do is take that acetyl group and dump it into a cyclic reaction cycle, or citric acid cycle, to produce our electron carriers. That's our NADH, FADH2. Those electron carriers then get moved on into our oxidative phosphorylation to produce our ATP. So we've got kind of this big long sequence. So the plan for this unit is to start with glucose. We move into acetyl-CoA, which they've got shown as this big thing. It's one reaction. It's a big one reaction, but it's one reaction, which then dumps into the citric acid cycle, which loops through. In the process of doing the citric acid cycle, we off-put electrons. Electrons are not stable, so they get dumped into our electron carriers, NADH and FADH2. Those electron carriers transport and dump their electrons into the electron transport chain, oftentimes abbreviated ETC. In the electron transport chain, the electrons get shuttled back and forth, okay, between different carriers to transfer what? Anybody remember biology? What are they doing in the electron transport chain? That's the end goal, is to release water. The whole point of the electron transport chain is to shuttle hydrogen ions across a membrane. Okay? Which means on one side of the membrane we become very acidic, and on the other side of the membrane we become, yeah, we could say basic. That works. Okay? Really what we're generating is an ion imbalance. So why do I care about an ion imbalance? Okay, how did you start your car this morning? Struck the key in the ignition, you turned it, right? Oh yeah, that's how we started it. Well, what's happening when you turn that ignition?
It's like an impulse flux.
To do what? Where's
Turn the engine over.
how is your engine getting turned over?
Battery.
The battery. Well, what's a battery?
Energy source.
Look at a battery. What do we have on it? We have a positive and a negative. What do we have in a battery? An ion imbalance. By controlling how those ions recombine, we can then generate electricity, and that electricity we can use for something. Guess what the electron transport chain is doing? The exact same thing. It is generating a battery. Once that battery has been built, we can then allow the recombination of those ions to form ATP. Okay. Where we use it for electrical energy, biology is using it for chemical energy. Kind of a neat little process. Okay. Your electron well, actually this is a nice little thing showing where all this is happening. Okay. I see two membranes. Where does that happen? Where do I have two membranes? What's that?
Our cells.
Yes, our cells, just floating around, happen normally in our cells. That means we're producing, remember we said a high acid side. So our cells are now stupidly acidic. What happens if we're stupidly acidic? We die. So this reaction needs to produce a stupidly acidic section. If it's just happening in our free-floating cells or even outside our cells, that becomes problematic. We're dead. So where do we do it? We dump it into a specialized compartment known as the mitochondria. The mitochondria now have a sequence of membranes. Why do we want the sequence of membranes? Yes, to control where all the pieces go. Really, it's about the acid. Okay? What happens if that membrane breaks? Dumps acid into the cell and we die, or the cell dies. So we want multiple membranes so that we can transfer the hydrogens across a membrane, but then not worry about the hydrogens going out into the rest of the cell. Okay? We're trying to keep everything localized as far as the chemistry is happening. Right? So pretty much everything we're talking about is happening in one localized environment. That's our mitochondria. The mitochondria, I really should remember to actually talk about cell structure, I guess, at the beginning of the semester. Anybody remember our cell structure for a mitochondria? It's the one organelle that has two membranes. Okay? It has an outer membrane and an inner membrane. Okay? All the other organelles, at best, have one membrane. Okay? So what we're talking about within this is where all of those molecules, where are the molecules of glycolysis? Where are those being moved? Remember, they need to eventually make it to the mitochondria, which means a lot of this process is happening in or around the mitochondria so that we can do simple transport back and forth. Okay? That said, where do I want the ATP? Everywhere. I want ATP everywhere. If ATP is only in the mitochondria, well then I don't have energy to do anything else, okay? Because it's only stuck to the mitochondria. Okay? So our molecules in each of these stages do have different processes and can branch outwards. Why would they branch outwards away from glycolysis or citric acid cycle? Why would pyruvate be used somewhere other than citric acid cycle or glycolysis? We could do something with anaerobic, that's a possibility. What is in those molecules?
CO2.
Carbon, oxygen, hydrogen, nitrogen. What is special about, say, elements of carbon, hydrogen, nitrogen, and oxygen? Are they anywhere else in the cell? Like DNA, like our proteins, like carbohydrates that are embedded in the cell walls so that we can recognize different blood molecules. Okay? We need those pieces. Those elements are a finite resource. So it's a question of where do those elements need to be. If they're in glycolysis and the citric acid cycle, why are they there and not somewhere else? What is the goal of citric acid and all of that? That's an indirect goal. Yes, electrons we need, but why are we generating electrons? To make energy. Okay? Why do we have all the molecules going through glycolysis, citric acid cycle, and all that fun stuff? We need energy. For whatever reason, the cell is in a situation where it needs energy. So what's going to happen? All of the resources are going to be dumped into the mitochondria. We aren't going to be looking at those molecules going out to do anything else. They're going to be involved centrally within this cycle to produce energy. What happens if the cell stops needing energy? That process needs to stop. But if I have all of the reagents to do that process, it's going to continue to go. So what should I do with those reagents? Get rid of them. Dump them somewhere else. Put them into, say, a protein. Put them into, say, DNA. Push those pieces outwards into the rest of the cell because those elements are a building block. Right? So it's not just this is happening all the time. This is happening when we need energy. If we don't need energy, all of the pieces leave. Right? Why is that a problem? If we're running the reaction, say, inside that inner membrane, how about in here? If I want those pieces to leave, what has to happen?
You have to cross the membrane.
They have to go through a membrane. Are they then in the cell? No. There's another membrane. So we have to transport between two membranes. Okay, what's the point of a membrane? Keep things separated. So to get these molecules outside of the membrane, we're also going to have transport mechanisms to get these things outside. Some of them will passively diffuse through. Most of them, or some important ones, require an active transport. We'll actually have to physically move them through the membrane. Okay? Enough side processes. Let's take a look at the citric acid cycle. This is another version of it. We start with pyruvate way up there at the top. We're trying to draw that structure. So it is kind of nice that we have the structures on here. It's a bit irritating that they're cartoony. We have our CH3. We have our carbonyl. And then we have the carboxylic acid of our pyruvate. So we're taking that pyruvate and we need to get acetyl-CoA. Well, the acetyl group is this half. So in that very first step, what do I need to do? I have to break a bond. I have to break the bond between those two carbons, which means what am I getting rid of? Sort of, not carboxylic acid, CO2. Okay? So we'll be off-gassing CO2 to get into the citric acid cycle. This is one of the reasons why we call it respiration. Okay? That's one of our byproducts. Okay? Once we've gotten the acetyl group in an active form attached to coenzyme A, now I can start to dump the acetyl group into the citric acid cycle. Okay? And so what I'm bringing in is 2 carbons and an oxygen. And I will react those 2 carbons and an oxygen with what molecule? So here's acetyl-CoA coming in. What do I react it with? Water is being reacted in there, yes. Citrate? Citrate's being reacted with those? Where is citrate? On the left or right side of those arrows? On the right side of those arrows. Where are your reactants typically? On the left side. So citrate is a product. What are you reacting the acetyl-CoA with? So as another option, we could go through and take a look at the structure of citrate. That has a whole lot more than 2 carbons and an oxygen coming from the acetyl group. Where did I get those pieces? The oxaloacetate. So I will react oxaloacetate with the acetyl-CoA to make citrate. So I'm synthesizing something. What would be a good enzyme name for that? Synthase. Oh, amazing. What am I making? Citrate. So we'll call it citrate synthase. Okay? So we're still responsible for those enzyme names and how they process through the structures of all of these nasty, nitpicky things. Okay? There isn't really anything I can do about the enzyme names. You're going to be responsible for those. The structure and the name of all of the reagents or all of the intermediates, I'll provide you structures and names and may ask for you to elaborate. Okay? So I would highly encourage you to memorize both, okay? Though memorization won't be strictly necessary because you're being asked to process what's happening at that point. Kind of makes sense? Okay. What happens to citrate? Citrate's here. What do we do to the citrate? What should we find? Another arrow. What's that arrow saying we do? We see an arrow going away from it, pointing to water, which means what did we do? We removed water. We did a dehydration reaction to form cis-aconitate. Okay. What enzyme did we use to do that? Aconitase. Not saying they're the nicest names. We at least know it's an enzyme because it says ACE. Okay, that's kind of nice. Okay. I find that one to be a bit of an odd structure to show because then what do we then do to cis-aconitate? We add water back to it. Okay, that's a bit weird. So we rehydrate it. Well, what enzyme do we use? The exact same enzyme, aconitase. So really what we're doing is converting citrate to D-isocitrate. Okay? And to do that, we use the enzyme aconitase. Well, that doesn't make any sense because aconitase is aconitate is an intermediate within that reaction. It is a stable, neutral-ish structure, and because of that, we show that structure. But it's still stuck within the same enzyme. So the aconitase enzyme removes water and then puts the water back on the structure. Did it put the water back in the exact same spot? No, because it has a different name. Okay, so we end up with a D-isocitrate. Okay. what we're trying to do is move around the cycle and see what each of those individual structures are, each of those names are, what the enzymes are, and are there important cofactors involved in each of those stages. The important cofactors to be aware of would be NADH, a redox mediator. So anything producing NADH or FADH2, anything involving your redox mediators, is going to be stupidly important because the redox mediators are used to make energy later on in our oxidative phosphorylation. Right? Everything that does not involve the redox mediators is trying to adjust the structure so that the redox mediators can actually work or function. Okay? If we look at this image, it is rather blurry and rather large and obnoxious. But I would argue it's got a little bit more information than this one. Right? Hey. Sorry, give me a sec. This is what I thought was popping up. So this is now the citric acid cycle with all the structures, the names of, I named that one wrong. The names of all of those structures. I've removed all of the redox mediators because I would argue the redox mediators you should be able to effectively derive by looking at the reaction, okay, which is a super common question, at least from my perspective. Okay. So looking at those structures, the names, and saying, okay, what things happened there. By looking at the structure, you can predict what was the change. Do I need a redox mediator? Do I not? Okay. So you don't have to memorize. Okay. If you want to memorize, instead of understanding what's happening at the structural level, that is fine. So what do I mean by that? Look at citrate to cis-aconitate. What changed? We're just going from here to here. We lost an oxygen, so that sounds like a redox reaction. What else did we lose? Hydrogen, which means lose an oxygen and you lose hydrogen. Losing oxygen is reduction. Losing hydrogen is oxidation. We've counteracted, which means, is this a reaction that requires a redox mediator? No. So no NADH there. How about cis-aconitate to deisocitrate? What changed? We added oxygen. Oh, so that's an oxidation reaction. But we also added hydrogen, which is reduction. Do we need NADH? No. Okay. How about deisocitrate to alpha ketoglutarate? That should be an alpha. I just screwed up that A. We lost CO2. And we added a bond to oxygen. So we did a redox reaction. Do I need NADH? Yes. Okay. So by looking at the structures, you can start to predict, do I need a redox mediator or not? Okay. That can at least set you up to figure out where things are produced by evaluating structure as opposed to memorization. Okay. Both work. The evaluation of a structure requires probably a heavier chemistry content on your fundamentals. Okay. So that depends how well you did in all of your chemistries, regardless of the path. If you did well in 130 and 230, evaluating the structure shouldn't be a problem. If you didn't do well, well, then you might want to just memorize, which is fine. Kind of makes sense? Okay. So what we will do in maybe Thursday, because I still got to go back to a couple slides, is look at each of those steps and say, what happened? What happened? What happened? What are the names of the enzymes being used? Okay. Did we need NAD? Do we produce NADH? Did we produce FADH2? Okay. Did we produce energy in a different format? How are each of those stages happening? And thinking at least a little bit about the mechanism. Yes, Anna.
So you know you need a redox mediator if you do something besides hydrogen.
When do we use a redox mediator? When you have a redox reaction. How do you know you have a redox reaction? change in the bonds to oxygen, change in the bonds to hydrogen. If you're doing both, you should be doing both in the same direction. Meaning if I decrease the bonds to oxygen, what do I also need to do?
Increase the bonds of hydrogen.
Increase the bonds to hydrogen, and now I've done a reduction. But if I decrease the bonds to oxygen and decrease the bonds to hydrogen, well, decreasing the bonds to oxygen is a reduction. Decreasing the bonds to hydrogen is oxidation. I've now paired two things that are opposites. I don't need a redox mediator because the electron transfer balances out. I don't need an extra piece. Does that make sense? Okay. So now let's backtrack a little bit here. We'll look at the exam questions in a second. Okay. So before we can even get into the citric acid cycle, we need to convert pyruvate into acetyl-CoA. We're trying to make that acetyl group something that can sort of easily transfer into the reaction. Okay? Which means that acetyl group, okay, the carbonyl carbon, needs to become very partially positive. My acetyl needs to be positive there. Okay? It is partially positive already because it's connected to carbon. Or because it's connected to the oxygen. Okay? But it can't be formally positive because it already has another bond to another carbon. Okay? That bond must break. But what kind of bond is that? That would be a carbon-carbon bond, also known as a nonpolar covalent bond, which happens to be a really strong bond. So that is not an easy bond to break off to get the acetyl group. So I need to do some crazy weird stuff to get that to happen, much like I did in the pyruvate decarboxylase when we looked at our anaerobic reaction. Except now we're using pyruvate dehydrogenase. What does the pyruvate dehydrogenase do? Well, it takes pyruvate, it removes that pesky carbon that I didn't want. It removes that piece. Okay? But it can't remove that piece easily, okay, for a couple reasons. If we draw out that structure, okay, to get CO2, what is that negative charge going to do? Donate electrons into the carbon. There's CO2, but what's the problem with that carbon? It breaks the octet, which means the electrons in the next bond have to go where? Which would make that carbon negative. That's a problem, because what does it need to be? What does it need to be, ultimately? What are we trying to get that carbon to be? Positive. Okay. If, let's say, we just try to do that on our own, is that structure stable? Why not? Carbon does not like to hold a negative charge, so it's too small, not high enough electronegativity. Is there resonance? Okay. No, because if I tried to push those electrons towards oxygen, I would exceed the octet on the oxygen. So there is no resonance to stabilize that. If there's no resonance to stabilize that, what does that mean about those mechanistic arrows? They are bad. They are invalid. We need something to help shuttle the electrons around to get that to occur. What is that something? Officially, the thiamine. The thiamine will act as an electron donator, which can help polarize that bond. Whoa, that was not what I wanted. Let's try that again. The thiamine, I'm just going to say THIA, can come in and help polarize that, okay, so that when the electrons from my CO2 come in, the electrons in that purple bond, instead of going to the carbon, will go to the thiamine. That can help cleave and separate those. The result is I end up with TPP with the acetyl group attached. Okay? The acetyl group is now an alcohol, though. Okay? I need the acetyl group back, right? Which means I need the electrons in that bond to come back over here and then to kick out that group, right? But then what would I make? Not quite.
Oh, the negative.
Would I have the acetyl? Sort of. I would have acetaldehyde, which would be awesome if this was the anaerobic process, because that now kills that group. I no longer have the acetyl reactivity anymore. It's now as an aldehyde, and I could process that into alcohol, produce NAD+, so that I could then go through glycolysis again. But that doesn't produce enough energy. So instead, what does our dehydrogenase complex do? Okay. It then shuttles the acetyl group to lipoic acid. Okay. The sulfur-sulfur bond, remember I said sulfur was a nasty reagent? Here it is. That sulfur-sulfur can go through and kick out the TPP. Please reconnect. And the process, we end up connecting the acetyl group now to that sulfur group. Okay? But I need to get the acetyl group onto something that can now easily transfer that isn't embedded inside a massive enzyme complex. Oh, that's where I can use a coenzyme. The coenzyme can now come in, pick up the acetyl group, and I now have my acetyl group attached to a mobile structure. The problem with this is I now don't have the sulfur-sulfur bond that I needed to refresh the cycle, so what has to happen? NAD+ comes in to finalize that and make NADH. Oh my God. Okay. So I am doing a redox reaction here. And I produce NADH in a massive enzyme complex. Okay? And that is pretty freaking sweet. Okay? Notice that they also add this extra little term FAD here. Anybody remember the difference between NAD and FAD? It's a harder one if you don't remember the structures. And I'm not about to flip all the way back to find them. FAD was relatively large in comparison to NAD. What does that mean about the mobility of FAD? Not very mobile. So as a cofactor, it gets dropped in and stuck kind of permanently within this enzyme complex. Okay? If I want that electron mediator to be mobile, I then need to dump electrons from the FAD out onto NAD. And I now have a more mobile version of my electrons on my NADH. Okay. That process is very, very similar to what we looked at already, and I think I moved the slides. Oh, never mind, I didn't move the slide. Okay. We've got our thiamine being shown in the upper left-hand corner. There's our biological carbene. That says car. Oh, screw it. Let's just write that again. Okay, that red carbon is our carbene because it can act in that very first step as a nucleophile. Okay. Which then sets us up for the very next step. Remember we said the lone pair on that oxygen comes in. Okay. We would then have to kick out electrons. Those electrons are coming out and being shared with our carbene carbon. And for sake of color coding, our electrons come in, shuttle out to the carbene carbon. It's now acting as effectively an electrophile by accepting electrons. Okay. We've now decarboxylated. This sets us up with our first intermediate. We now have to reverse that process. The rest of that is now how we reverse it. Okay. We get darn close to the end here, to fully cleave the structure off, that's where our lipoic acid comes in to react out. Eventually the lipoic acid goes around, cyclizes back, and we get our coenzyme A, okay, or our acetyl coenzyme A. Kind of a neat process. Okay? The be-all, end-all of this is what's in the lower right-hand corner. This is what most biochemists go through and say, oh, I want something to happen, the enzyme did it. Okay? I don't need to worry about how it happened, I just need to know that it happened. Okay? Because the stuff happening in there is really detailed. Okay? And to nail that all down, we've got a lot of work to go through. Okay? It's also where it's interesting, at least to me. So with that, let's take a look at some enzyme questions. By enzyme, I mean exam. What role does thiamine play in many redox reactions? It's actually kind of a hard question. It was a carbene, right? What did a carbene act as? An electrophile and a nucleophile. These aren't strictly multiple choice because I can do whatever I want with them. So both answer A and B are true. Okay. We could arguably go through and say D was true also, because for it to act as a nucleophile, what had to happen? So if we go all the way back, our thiamine is our upper left-hand corner. That's its active form. That's supercharged. Remember, our thiamine actually starts protonated with the hydrogen. So to get the active form, what has to happen? It has to donate a hydrogen, so it acts as an acid to go through and form the active nucleophile. That can then act as an electrophile and accept electrons later on. Okay. Thiamine is an example of a naturally stable carbene. Does the conversion from ADP to ATP require oxidation? This would be a much trickier question if I'd actually asked this significantly later in the slides, after we talked about electron transport chain. Why are you saying no, Mark?
Vaguely, I don't remember anything. It's just, I don't know how to explain it, that's just what.
It's a phosphorylation reaction. Does that require oxidation?
That's what I was trying.
When does it not require oxidation? As a hint, we just walked through the steps on when it doesn't require oxidation. We just finished it. Probably Thursday is actually when we officially finished it. No, fermentation actually does not produce ATP. It produces NAD+. Glycolysis. Glycolysis produces ATP. There's no oxidation. Okay? We don't need to do oxidation to generate ATP. Okay? Arguably, ATP synthase isn't doing oxidation. It, as an enzyme, doesn't do oxidation. For ATP synthase to function, we have to have a proton gradient, which is made using oxidation, which is why ATP synthase is typically connected to oxidative phosphorylation. Okay? Remember, we had substrate level phosphorylation and oxidative phosphorylation. Substrate level does not require oxidation. That's why it's called substrate level and not oxidative phosphorylation. Okay? Which of the following does not contain enolizable hydrogens? Okay, which may look really, really weird, so let's piece some of this out, because we actually talked about this enough that I think we talked about it enough. I may have to go back and watch the videos, but I'm pretty sure we talked about it. Let's clean this up and just look at enol. What is the enol? What is an alkene? Carbon-carbon pi bond. What is an ol?
Alcohol.
An alcohol. That structure is not stable. Remember I said that this was one of the most important molecules in OChem that should have been addressed very, very heavily in OChem and borderline isn't. That structure can do what is known as a tautomerization, which I don't think I said before, to make a ketone. Right, we add an extra carbon there. So what happened when I convert between the ketone form and the enol form? What did I do? Dealing with your oxygen is probably a bad idea. Broke the carbon-carbon pi bond by doing what? Unfortunately, no. So a lot of people like to say that this is happening. That doesn't make sense because then the oxygen exceeds its octet and it'd have to be kicking out H-. That's really bad.
Does it lose hydrogen?
That hydrogen is relatively acidic, so the electrons actually come this way, which means the electrons in that pi bond need to come out to pick up a hydrogen. What charge should that hydrogen be? Positively charged. What charge was the purple hydrogen? Positively charged. What are we doing in the course of this reaction? We are changing the location of the hydrogen from being attached to the oxygen to being attached to the carbon. Okay. Where's that carbon relative to the oxygen? Okay, so we could call it as 1, 2. Okay. Arguably the carbon or the oxygen is part of that carbon as a carbonyl at position 1. So that carbon is actually arguably 0, which then means the next carbon would be carbon number 1. But that can be confusing if we number it. So instead of numbering it, we use Greek letters and we call it the alpha carbon. So what do we need to find? Well, we need a carbonyl. What else do we need? An alpha carbon. What has to be at the alpha carbon? A hydrogen. If there is no hydrogen at the alpha carbon, then I can't remove that hydrogen to put it on the oxygen. Okay, so which of those structures have an alpha hydrogen? Okay, there's my carbonyl. I look at the alpha position, it's a sulfur. That doesn't count. If I go to the alpha or the other direction, what do I have out there? There's an alpha hydrogen, which means that structure can enolize. That one counts. What about the next one? There's my carbonyl. Look at the alpha carbon. Do I have hydrogens there? Yes. Guess what that one is? Enolizable. Look at the next structure. There's no carbonyl, which means can't enolize. Next one. Right. We have the carbonyl, and we have a hydrogen.
No carbon.
It needs to be attached to a carbon. We need to have the alpha carbon. Right? So there's no carbon that direction, there's no carbon the other way, and then if we look out the other end, no carbon out there. Right? Finding an enolizable hydrogen is important. Right? One of those molecules is biologically relevant. Which one is that? The first one is biologically relevant because what do we have? We have acetyl-CoA. How is that molecule reactive? Through the alpha hydrogen. Right? And we'll see that in just probably not today. Okay? But that hydrogen can get removed, making the enolate. Okay? And really what we're saying is it can be removed and we can stabilize that negative carbon. What do I now have? I have a nucleophile. That carbon can then go through and do some chemistry. Sweet. Why might it be important for citrate synthase to be allosterically affected by NADH Yeah, it's not a really fun question at this point. Okay, so on the test, there'd be a subtle change to this. What would that change be? I would provide the reaction for citrate synthase. Okay, which, do we have it on the next slide? There it is. Citrate synthase is this one. Okay. Why do I want that reaction to be controlled by NADH?
You're saying then we want the redox reaction.
I'm not saying that necessarily.
Isn't that why NADH is there?
Is acetyl-CoA part of the citric acid cycle?
It gets dumped into the
It gets dumped into the citric acid cycle. Pyruvate is an awesome, or sorry, acetyl-CoA is an awesome molecule because it can transfer acetyl groups to a variety of different things. Okay, so it's synthetically awesome. That's like it's almost the equivalent of ATP for synthesis. I can now dump 2 carbons and an oxygen on pretty much any structure. Okay, that's pretty useful. Okay. If I dump it into the citric acid cycle, what am I now committed to? The whole process of the citric acid cycle, which means I'm making energy. Why do I want that reaction to be controlled? I'm taking an insanely useful synthetic molecule and I'm saying, make me energy. What if I don't want energy? Right? I want to stop that reaction and use the synthetically useful molecule somewhere else where it can be useful. I don't want to dump it into the cycle to produce more energy. So what we're asking about is that step. Right? Why should it be important to be controlled by NADH? Well, the whole goal of citric acid cycle is to produce NADH. If I've got a bunch of NADH, I don't want to make more. So NADH will come in and shut down citrate synthase to prevent the formation of more. What kind of? It's going to inhibit it. Okay? I want to shut down that reaction and prevent it from happening. The acetyl-CoA can then be used somewhere else. Okay? So when we're looking, like remember back to glycolysis, we had to find different inhibition sites. The same thing is going to happen in the citric acid cycle. We'll look at each of those pieces and say, is it important to shut this one down or not? Okay? If we go through and look at citrate synthase, do you see any NADH? Interesting. NAD plus and NADH are not used in that reaction. Okay? That becomes a harder one to nail down and see because we've got lots of pieces. You've got water, you've got acetyl-CoA, you've got oxaloacetate, and then you're also releasing the coenzyme A and citrate. Okay? So NAD is actually not used in that reaction. Okay? But it generates citrate, which then can be used to make NADH. Okay? So acetyl-CoA, super useful. Don't get rid of it or don't dump it into a cycle to produce things that you already have tons of. Okay? So it is going to be controlled based off our NADH. Kind of make sense? Okay. Maybe we'll just end there. The on-ramp to get in, okay, it is an exothermic reaction, which means it will happen on its own. We don't need to add energy into it. It will release energy technically. The amount of energy it releases is roughly equivalent to a single formation of ATP, but it doesn't have enough excess energy to produce more than that, or to produce an ATP. Your ATP is roughly, I think, 32 kilojoules. Well, that's 32.8, but we have to over-potential it to actually get the reaction to work. Okay? So we've got our structures. What we'll do when we pick up on Thursday is decide how and where that reaction occurs. So you'll look at the differences between your oxaloacetate and the citrate. and decide how the acetyl-CoA gets involved.